Here in my lab, we have a group of biochemistry researchers, and we're really interested in the biology and biochemistry of a small bacterium called Mycobacterium tuberculosis. This is the bacterium that infects humans and causes the disease tuberculosis. Globally, it still is one of the most important killers of humans. It kills between 1.5 and 2 million people every year. So we here are interested in the biochemistry of proteins that perform particular functions in the bacterium. It's a terrible pathogen, but it's a very interesting pathogen because it lives with humans, in most cases, without symptoms. So it's estimated that about a third of the world's population carry the bacterium. The bacterium lives very happily in humans without causing any problems. And the reason it is able to do that is because it effectively can lie dormant. And this is called latent tuberculosis. And so the way that the bacterium can do this is of particular interest. One of the other issues is that this latent bacterium can resist antibiotic treatment because normal antibiotics treat actively growing bacteria. How does the bacterium maintain its metabolism at such a low level? So essentially the bacterium has shut down its growth, and in shutting down its growth it's able to avoid the immune system, and it's also able to resist antibiotic treatment. And so along with my team here, we work on what we would describe as metabolic control by the bacterium. The bug gets under stress, and then its reaction is to slow down its metabolism. We're interested in the biochemistry of that process. So there are several strains that have been circulating in New Zealand for at least the last 30 years, and we've been unable to get rid of them. About 25% of cases in New Zealand are caused by 3 strains: the Rangipo strain, the Southern Cross strain, and the Otara strain. And we're really interested in why they persist in New Zealand, why we can't get rid of them, and why these strains are different from strains that come in from overseas. And one of the ways that you can look at this is that you can sequence the genomes of these bugs, and then you can compare these strains and try to decipher A, where they came from, and B, what the differences are that allow these strains to persist in New Zealand.